Our next presenter for the session is now Dr. Sarah Hedrich. Dr. Hedrich, you're welcome to start sharing your slides when you're ready. Dr. Hedrich obtained her PhD in pharmacology and toxicology from the Freie University in Berlin, Germany in 2009. She has since done research at the Ludwig Maximilians University in Munich, Tufts University in Boston, and again in Berlin. She is now an assistant professor at the University of British Columbia, and her research focuses on inflammatory and genetic diseases of human epithelial cells, the development of novel therapeutic approaches, and tissue engineering of complex human-based organ models aiming for valid and predictive test systems for preclinical and fundamental research. Her talk today is titled, How COVID Spotlights the Model Dilemma in Biomedical Research. Welcome, Dr. Hedrich, and I'll now turn the presentation over to you.
Thank you very much, Kate, for the introduction. And
Dr. Hedrich, we are actually looking at your presenter mode on your screen. So if you just go up and on display settings on that page as well. Nope.
You know what? I think I know.
Do you have two screens?
Yeah, I have two screens. Sorry, I always forget about that.
That's okay.
Just a sec. I know how to resolve that. Does it work
perfect
now?
Yes, okay. Perfect. Thank you. Sorry for the interruption.
Yeah, no worries. Yeah, as I said, it's a great pleasure for me to be here. And as just introduced by Kate, my talk is about how COVID spotlights the model dilemma in biomedical research. And some of you now may ask, what does the model dilemma in biomedical research has to do with COVID? So let's take a step back and look at the facts that we know, at very basic facts that we know about COVID. So COVID-19 is a zoonotic disease. And at the beginning of every new zoonotic disease, or after the discovery of a new pathogenic virus or microbe in general, there are three key questions that we need to ask ourselves, or that need to be answered as fast as possible. Number one is, what is the causative agent? Number two is, what are the hosts, or what is the host? And number three is, what are the intermediate hosts? And although we were able to fairly quickly question number one and number two, there's still some ambiguity around question number three. So what are the intermediate hosts? And at the beginning of a new pandemic, and after the discovery of a new human pathogenic microbe or virus, we always have the same problem. And the problem is that there are no established models available. And so what happened also at this pandemic and previously, and what's also continuously happening in biomedical research, is that we reflexively look for animal models. And just to be honest, that always costs us a lot of time. And in case of COVID-19, it costs a lot of valuable time, a lot of money, which is needed for the development of such animal models. Nonetheless, what the gold standard for COVID-19, and to better understand what the virus is doing and how we can treat those infections, or potentially even prevent the spread and the infection itself, the gold standard we are looking at is the human being. And so I think a very important question that we need to answer, that we need to ask ourselves is, wouldn't it be smarter to develop human-based models, or to utilize human-based models instead of animal models? And this is the key question that I'm trying to, or that I will allude to over the course of my presentation. And this is also the question that we discussed in a recent article that was published that I published with a couple of colleagues. These are my students, and this is a colleague from Berlin in Nature Reviews Materials. And when we look at what COVID does, we know that COVID is caused by the SARS-CoV-2 virus. And we also know that the SARS-CoV-2 virus requires the expression of ACE2, or requires the ACE2 receptor for cellular entry. We also know that the expression levels of a protease, TMPRSS2, is critical for cellular entry as well. And unfortunately, we are also well aware that COVID-19 is a disease that is not restricted to the lung, but that in some cases, and still today, we cannot surely and entirely predict in what cases or in what patients that will happen. It can also spread to other organs, it can affect other organs, and it can cause multi-organ failure, which very often then also results in the death of the patient. Coming back to the COVID-19 models, at the beginning, as I said, at the beginning of the COVID-19 pandemic, there was no established model. And back in summer last year, the WHO formed an expert panel in order to identify the most suitable animal model for COVID-19. And this expert panel published their results, I think, in fall last year. And what they concluded is that there, in fact, is no animal model that fully emulates human COVID-19. And what you see here, I mean, we already heard the previous talk. There was the presentation about the hamster. This is the golden Syrian hamster. These are the ferrets. These are fairly commonly used COVID-19 models, because they have, they are susceptible to the disease. They have they express ACE2 receptors and the required protease. However, what we need to keep in mind that the expression levels of those proteins can be quite distinct from the human situation. Mice in general are not susceptible to SARS-CoV-2 virus, but after genetic manipulation, rodents can also be used as COVID-19 model. But what is often observed, or in some cases observed in mice, for example, is a distorted pathogenicity, which means that mice develop an encephalitis, which as such is not observed in human COVID-19. And in fact, this is also a pathogenicity that has been previously reported for SARS and MERS, so other coronaviruses as well. Another limitation is also that dependent on the mouse model and other models, animal models as well, we either get a pretty mild or a very severe course of disease. And this automatically means that by choosing a specific animal model, we automatically generate a bias in what type of disease or type of COVID-19 infection we are investigating, and the results we will be able to retrieve from that as well. And the other limitation is, or the other caveat is that there are not many reagents and consumables that go well with ferrets, for example, ferret tissue or golden Syrian hamster tissue. So, the limited access or the limited availability of species-specific reagents is also a great limitation, which hampers the research in animal models as well. There are a couple of human-based COVID-19 models, and I start with a very simplistic model, which is clearly the 2D cell culture. 2D cell culture models are widely used. They are very important and very easy to use models, cell lines, but also primary cells. And I would like to emphasize that primary cells are clearly the model of choice. There's always the discussion about limited accessibility, although currently, as of today, there are quite a few of companies that provide access, but there are also biobanks through which you can access primary cell cultures. Nonetheless, this is an important tool, but also here, what you need to keep in mind is that the results that you will get strongly depend on the expression levels of for example, the ACE2 receptor or the TMPRSS2 protease. And so, by choosing a specific cell line, this automatically also generates a bias and can skew results in one way or the other. A little more complex and especially three-dimensional models are organoids and three classical air-liquid interface models like the 3D tissue models, and they have been pretty commonly used over the past year to study virus-host interactions and also for drug screening and drug repurposing studies. And in fact, there have been a couple of drugs that prove very efficient in organoids that have now entered clinical trials and are currently undergoing clinical trials with respect to COVID-19. And I would also like to emphasize that there is a nice head-to-head comparison where the results that were derived from organoids have been also received from mouse models, which clearly shows that organoids, or those three-dimensional models, can be standalone models when it comes to COVID-19 research. Nonetheless, obvious limitation of organoids and the three-dimensional tissue models is that these are pretty simplistic models, and they don't have an organ, so they are not connected. There is no systemic circulation, and they are not connected with other tissues. And so I really think that, the future in biomedical research in general, but also when we think about potential future pandemics, that we need to really increase the efforts in the development and utilization of organ and humans on a chip. And I will come back to that later in my talk, because the great benefit and advantage of those organ or human on a chip setups is that you can co-cultivate several organ models, complex three-dimensional tissue models, that those tissue models are connected through microfluidic channels, and those channels, or this chip-like setup, is then connected to a pump, and this pump ensures and can arrange that the cell culture medium is, continuously flows, and it flows at a velocity which is very comparable to in vivo, actually, which means that using organs on a chip or human on a chip setups, that we get pretty close to human physiology and potentially also pathophysiology. So coming back to one of my initial questions, wouldn't it make more sense to develop human-based models? And I think it's a clear yes. So yes, we must develop, it's crucial, we must develop human-based models, also to be able to respond better to the next pandemic, to the next emerging virus, so that, the next time a virus hits, we don't need to scramble and need to look for, okay, what is a suitable model? Because the gold standard, and that's something we need to keep in mind, is the gold standard is the human being. But the model dilemma is clearly not COVID specific. COVID just highlighted it once again. And this is actually a slide from a talk that I attended a couple of weeks back. And this talk was given by Linda Griffith from MIT. And she does a lot of research in endometriosis. And a couple of years back, her group discovered a pathway which involved Janus kinases as key mediators and drivers of endometriosis. And based on those findings, Janus kinase inhibitors have been developed, and they have been tested in a great variety of patient populations, like mice and also in monkeys. And they were pretty effective in mice and monkeys, but unfortunately, they were not effective at all in humans. And so this basically was one trigger for her to move away from the animal models and to move towards human-based systems, because there is a clear interspecies related difference. And in order to overcome that, we really need to focus and we need to emphasize our efforts in developing human-based tissue models. And I just wanted to show that because I think this is an excellent example for why it is important to move towards human-based approaches. So I'm in pharmaceutical sciences. My background is also, I'm a pharmacist by training, in fact. And another aspect that I want to highlight here is that when we look at the drug development process in general, I think we all know that this is a highly inefficient process. So the development costs of drugs are disproportionately high, whereas on the other hand, the drug attrition rate over the course of preclinical phase and then clinical phase is extremely high. And it has been about 90 to 95%. It also depends on the indication we are looking at, but the drug attrition rate has been that high for decades, to be fair. And there are a couple of reasons for that, and it's not all about the animal models and the model dilemma I'm alluding to, but it is definitely one reason. And as I said, there are a couple of reasons. There are, certain caveats in the clinical trial design. There are method-related deficits. And what I mean with that is really the preclinical models. And I will discuss that in more detail in a few seconds. But there are often also regulatory and economical reasons for this high drug attrition rate through the drug development process. But when we look at the preclinical research, there are many flaws with respect to the preclinical study design. So one aspect, and I already mentioned that repeatedly, there are distinct interspecies related differences. A mouse is not a human being. Nonetheless, there's often also a lack of suitable models. So, there are some of the human diseases cannot be simulated or emulated well in animal models. Just to give you one example, my research focuses on atopic diseases. And there are several atopic dermatitis mouse models out there. The problem is, however, that mice do not spontaneously develop atopic dermatitis. So what we do is we highly artificially induce a phenotype that somehow looks similar to what we see in human atopic dermatitis. And then we use this model to study pathophysiology, and then we use this model also to test the efficacy and safety of drug candidates. And it's obvious that there is a disconnect because nobody knows. And in fact, we now know over the course of the past years that many animal models are not predictive or not well predictive of the human situation. And that's just something we have to be cautious about. And also the study design of preclinical research, low case sizes or low case numbers, and also lack of heterogeneity. Just to mention one example, there are still a couple of researchers out that when they conduct animal research or animal studies, that all of the animals that are being used are in fact male, and there's very strong. It's crystal clear that there are sex-related differences between men and females. And so by not including female mice into a preclinical study, we completely ignore 50% of the population when it comes then to the human population. And this is a clear flaw in preclinical study design. There's another good reason why we urgently need human-based models, because we are in the phase of modern drugs. And what I mean with modern drugs are things like biologics, antibodies. And in fact, almost half of the drugs that are currently undergoing development or that are in the drug development pipeline are biologics. And the majority of those biologicals, they only recognize human targets, which means that if I want to test the efficacy and the safety of those biologicals, if I do that in an animal model, I need to modify the biological sometimes quite significantly in order to make it, usable in an animal-based setting. And that doesn't make sense because we want to, the ultimate goal is to treat a human being for a disease, at least when we talk about drugs for humans. And so why are we still taking the step back? We should take a step forward towards human-based models. And another example I want to give you is here, gene therapy. I mean, I'm pretty sure all of you have heard about CRISPR-Cas. And CRISPR-Cas is also species specific. What happens with CRISPR-Cas is that you have the gene scissor, the Cas enzyme, and then you have a guide RNA. And this guide RNA is a sequence which depends on the gene sequence. And this guide RNA directs the Cas9 protein, so the gene scissor, to the target area. But the genetic code differs between species, and also the genetic code encodes for different things when I look at a mouse or a human being. So this also again means that when I test CRISPR-Cas in a rodent, for example, I need a different setup, I need a different system. And then again, the question is this really translatable to the human situation? As I mentioned previously, my lab is very much interested in studying inflammatory and genetic diseases of human epithelia. And we focus mainly on skin and on lung. And I just want to give you a few, a few other examples about the model dilemma my lab especially is continuously facing in our daily lives. So just by looking at how human skin looks like versus murine skin, it becomes obvious that there is a significant difference, and you don't have to be a skin expert for that. So this is human skin, this is a cross-section of human skin, this is a cross-section of mouse skin. And what becomes obvious, this is like the outermost layer, the so-called epidermis. And in mouse skin, this usually is only 1 to 3 epidermal layers. Whereas in human skin, we are looking at 15 to 20 epidermal layers, which means that the skin barrier function automatically is also significantly stronger in human skin and than in mouse skin. And there are a couple of publications in high-ranking journals out there that nicely show the delivery of XYZ into murine skin, and then including CRISPR-Cas, for example. The problem is that doesn't say anything about what it will look like in a human being or in a human setup, because mouse skin, it's so much easier to deliver stuff into the mouse skin or through the mouse skin because the barrier is significantly weaker and it's significantly thinner. The other differences are humans only have few hair follicles, mice have fur. Then there is this muscle layer in rodents, which is important for wound healing, for example. Human skin does not have this muscle layer, so the wound healing process is completely different between mice and humans. But still, rodents are widely used for wound healing studies. But just keep in mind, the wound healing process is completely different. The regenerative potential in mice is significantly higher. Hardly any scarring occurs in rodents. And this is important, especially for my research. So there are clear immunological differences between man and mice. And you cannot simply extrapolate that. And in fact, a nice study from 2014 showed that there's only 30% overlap of skin-associated genes between men and mice. So how are we trying to overcome those interspecies related differences? We are creating our own skin models. So we are exclusively working with primary cells. Skin relevant cells are keratinocytes and fibroblasts. We isolate them from excised skin that is derived from plastic surgeries. And then we have a transfer setup in which we can bring together, build together, stack together those cells again, and then we cultivate them at the air-liquid interface for 2 weeks approximately, and then the cells nicely differentiate and form a three-dimensional structure. And I hope you can see it okay, but this is just a cross-section of one of our skin models, and this is a cross-section of native human skin. And just keep in mind, I hope you still remember what mouse skin looked like. So look at this in comparison to this, and now look at this in comparison to this. So obviously our self-made human skin much better resembles actual human skin. And now you may ask, okay, what about diseases? And we can clearly emulate diseases in those three-dimensional tissue models. So we can, for example, modulate gene expression similar to what is being done in mice. So we can knock down or knock out disease-associated genes. We also work with patient-derived cells. We then bring them together and we can regrow three-dimensional skin, and it nicely represents the phenotype of the disease we are looking at. And we can also emulate inflammatory conditions. There are different ways to do that. For example, you can directly supplement disease-relevant cytokines. We are interested in atopic dermatitis, so we are mainly focusing on interleukin-4 and 13. And there's a lot of work going into the direction of immune cell implementation. I need to say that this is still a caveat of those three-dimensional models, because the most important immune cells when it comes to skin inflammation or not, maybe not the most important, but the immune cells that have a key role are dendritic cells. And so far, there have been no successful attempts to stably incorporate dendritic cells into three-dimensional tissue models for an extended period of time. But what works quite well is, for example, T-cell supplementation. And we have utilized that for several studies in our lab for the testing of drugs, but also to investigate disease-related mechanism. But the models are not complex enough. And I just briefly alluded to that already. And this is one of the main criticism that people like I hear all the time. And I just want to give you one example, and I could provide you many more for that, although they are not very complex, and I totally acknowledge that they are a simplified system, but sometimes I think in some cases we overestimate how important complexity is to answer specific questions. So one example I want to give you is a project from my group. So we looked at the skin pH, and in atopic dermatitis, the skin pH is unphysiological high. So we are looking at 6 to 6.5. The physiological skin pH is around 5.5. And we also know for atopic dermatitis that if someone has a mutation in a gene, which is called filaggrin gene, this predisposes this person for atopic dermatitis. And it is also known that this filaggrin protein is degraded in skin into organic acids. And so when you look into the textbooks and also in publications, you find that for the regulation of the skin pH, 3 main mechanisms are responsible. Number 1 is the degradation of filaggrin to organic acids. Number 2 is the generation of free fatty acids. And number 3 is the expression of a proton pump, NHE1. And when you look in the same publications, you find that there is the explanation for the unphysiologically high pH in atopic dermatitis is because filaggrin is missing, therefore no organic acids are formed. That's the reason for pH 6 and 6.5. But nobody really investigated that. And so what we did is we generated filaggrin deficient skin models, and we actually measured the pH. And what we found is that our control models, as well as our filaggrin deficient skin models, have a nicely physiological pH around 5.5. And then we looked further into that. And what we also found then is that obviously there is a feedback mechanism in skin. As soon as the skin senses that one pathway or one factor is missing, in that case filaggrin, it ramps up the other 2 mechanisms. So it increases the generation of free fatty acids, and it ramps up the expression of proton pump NHE1. And I'm pretty sure that this type of feedback mechanism would have not been identified in an animal model, because the animal models are in fact very complex. And, there are many different layers that overlay each other's. And so, those singularized aspects are very difficult to identify in very complex systems. And so with that study, we were able to show that filaggrin is not essential for skin pH. In consecutive studies, actually, we were then able to show that as soon as the inflammation kicks in, as soon as there is interleukin-4 and 13, which are relevant for atopic dermatitis, then we see the upregulation of this pH. And what we assume is that, the inflammation then counterbalances the feedback, or, counteracts the feedback mechanism, so that this is not able to compensate anymore. And then we see this unphysiologically high skin surface pH. But this needs to be verified in an animal model. This is one of my favorite comments when I submit grants or when I submit publications. This is a comment which I always get in reviews from publications. And is this really necessary? Because what is the rationale for that? It's the gold standard. So we have done animal work for decades. And so, now we are approaching the next step. We are approaching the human-based models, or we are utilizing human-based models. And again, the gold standard is the human being. So if the authors or the applicants, when it comes to grant proposals, if they can provide good rationales why it does not make sense to go into an animal, then I think this needs to be acknowledged and this needs to be accepted. Because there, as I said, there is no, especially with this pH story, there would have been no way that we could verify that in a mouse model. Mouse skin has a different pH, for example. There are different mechanisms and pathways involved. So there is no point at some, I'm not saying this is 100%. So I'm not saying that we can avoid all animal models, not at all, but there are clear limitations of animal models, and we definitely need human-based models in order to fill those gaps and to better bridge preclinical and clinical research. And I already mentioned that the main point of critique when it comes to human-based models is they don't have an immune system, which is true. And there's the lack of organ crosstalk, which is also true for the models that I just presented. And this brings me back to something that I mentioned a few slides back, which is the organs on a chip setups or the humans on a chip setup. And this is the future, and I really believe in that. I already briefly described how those organ on a chip setups work. So you can see one example here. There are a couple out there. They are also commercially available. Basically, what you can do is you can combine different three-dimensional tissue models on those organs on a chip setups, and then you connect it to a pump, and then you can emulate a systemic circulation. The commercially available systems currently allow a co-cultivation of up to 4 organs. But there are 2 US-based groups that have shown that it is possible to co-cultivate up to 10 different organs over an extended period of time. And as soon as we talk about co-cultivation of 10 different organ models or systems, tissues, then we are approaching the human on a chip. Because it is believed that as soon as we can combine the 10 organ systems or tissues on a chip, that this is sufficient complexity to emulate the organism of a living human being. And this is clearly the future of biomedical research. And this is one of my last slides. I just want to show you or give you an example how powerful those organ on a chip setups are. And this is a study that was conducted by Don Ingber's group at the Weiss Institute in Boston. And what they did, they connected a gut chip, a liver chip, and also a kidney chip. And what they wanted to do, or what they wanted to investigate, are the pharmacokinetics of approved drugs. And what they did is they tested they predicted the human pharmacokinetic parameters for different drugs. In that case, it was oral nicotine. So they looked at the nicotine gum. And then here on the right-hand side, you see the data derived from the organ on a chip setup, the PK data. And here in the middle, you see the PK data derived from clinical trial data. And when you look through this list, you clearly see that the PK values predicted by the multi-organ chips are identical or very close to what has been found in clinical data, clinical trials. And this clearly highlights how well and how predictive those in vitro systems, those organ on a chip systems, in fact, are. And a similar thing has been done with cisplatin, which is a widely used anti-cancer drug. Here are also the PK parameters shown in black. That's black is after 1 hour, and in blue, this is after 3 hours. The dotted line, this is the data derived from multi-organ chips. And the continuous line, these are the patient-derived data. And you can clearly see that there is almost 100% overlap between the data from the multi-organ chip and the actual patient data, which again shows how powerful those organ on a chip setups can be. And now I'm almost at the end, and I want to go back to the COVID-19 with what I started. I think that is exactly what we need to do for COVID-19 research, although it may be already a little bit too late, but in order to be better prepared for the next emerging virus. What we need is modular systems, we need simple systems, And we need definitely organ-on-a-chip setups that can be combined in a, in a plug-and-play setting, basically, so that we can, for example, connect brain tissue with lung tissue, with liver tissue, gut tissue, and kidney tissue, and then run infection studies with that. And there's still a long way ahead of us. I mentioned that there are a couple of those organ-on-a-chip setups are commercially available, but they are pretty stiff and they are pretty expensive setups, and most of them are not modular at all. So this is clearly something we have to overcome in the future in order to be prepared, to be better prepared in the future. But I'm convinced this is really the future, not only for COVID, but also for the entire biomedical research. With that, I'm at the end of my presentation. Thank you very much, and I'm happy to take any questions.